Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I'm Dr. Samhani. Sorry because tomorrow I cannot make it to have synchronous lecture with you. So I am recording my slides tonight. Inshallah, I won't go very long. So our topic is functional structures of the kidney and the renal circulation. So we have 4 learning objectives. Describe the physiological functions of the kidney, describe the types and structure of the nephron, describe the microstructure of the filtration barrier, and describe the renal circulation and renal blood flow through the nephron. So kidney functions. Kidney plays an important role in homeostatic functions of the body. Water, mineral, nutrient and gases pass through the ECF before entering the cellular compartment, and cellular waste products pass through the ECF before being excreted from body. Hence, maintenance of ECF volume and composition is vital for organ functions, and kidney plays important role in this homeostasis. Functions of kidney. There are many functions that played by kidney: urine formation and excretion of waste product, regulation of ECF volume, regulation of blood pressure, regulation of plasma osmolality, acid-base balance, regulation of electrolyte composition, regulation of erythropoiesis, endocrine function, and also gluconeogenesis. For formation and excretion of waste product, the primary function of the kidneys is to excrete waste products of metabolism from the body, such as urea, creatinine, uric acid, which dissolve in urine. Kidneys form urine and excrete many toxic waste products from the body, which dissolve in urine. Thus, kidneys prevent the accumulation to a dangerous level in the body. For regulation of ECF volume, kidneys play an important role in regulation of ECF volume of the body. Many hormones such as aldosterone, ADH, angiotensin involved in regulation of ECF volume act on kidney tubules to achieve this function. These hormones modify reabsorption of water and sodium from kidney to control ECF volume. Blood pressure. Kidney contributes mainly to long-term regulation of arterial volume and pressure. Renin, for example, secreted from kidney activates renin-angiotensin-aldosterone axis that plays an important role in regulation of BP. Blood pressure. Many hormones act on kidney to regulate blood volume and pressure. And kidney have their intrinsic mechanism to alter sodium and water excretion to regulate blood pressure. Regulation of electrolyte composition of body fluids is the main function of kidney. Excretion and reabsorption of electrolytes from kidney directly influence their concentration in ECF. Kidneys also contribute to vitamin D synthesis that control plasma calcium concentration. Acid-base balance. Kidneys contribute significantly to acid-base balance by controlling bicarbonate excretion and hydrogen ion secretion. Thus, it controls blood pH and pH of other body fluids. Therefore, kidney abnormalities may cause metabolic acidosis or alkalosis. Regulation of plasma osmolality. By controlling sodium and water reabsorption, kidneys control plasma osmolality. Change in plasma osmolality provides feedback signal for secretion of ADH that acts on kidney to regulate osmolality. So there are others like erythropoiesis, endocrine and gluconeogenesis. And we also have here diagram for water balance. When there is insufficient water in our body, so it will increase the blood osmolality. Because regulation of water is depends on water plasma osmolality. Osmolality is the ratio of solutes in solution to a volume of solvent in a solution. So plasma osmolality is thus the ratio of solutes to water in blood plasma. As the blood becomes more concentrated, the thirst response, which is a sequence of physiological process, is triggered. So there are the functions of osmoreceptor. Osmoreceptors are sensory receptors in the hypothalamus that monitor the concentration of solutes. If blood osmolality increases above its ideal value, the hypothalamus transmits signals that result in conscious awareness of thirst. The hypothalamus also releases antidiuretic hormone through the posterior pituitary gland. ADH signals the kidney to recover water from urine, effectively diluting the blood plasma. And there's also the mechanism where the mouth get dry. So this is to conserve water. The hypothalamus also sends signals via the sympathetic nervous system to our salivary glands in the mouth. The signals result in decrease in watery serous output. These changes in secretions result in dry mouth and sensation of thirst. Decreased blood volume also happen, resulting from water loss. So the heart is ultimately signaled to increase its rate to compensate the lowered blood pressure. Besides, angiotensin 2 will be increased to simulate thirst. Hence, fluid will be taken and blood osmolality will be decreased. So this is electrolyte balance. Regulation of blood calcium level. Okay, the blood calcium levels are regulated by parathyroid hormone, PTH, which is produced by the parathyroid glands. PTH is released in response to low blood calcium level. It increases level of calcium by targeting the skeleton, kidneys, and the intestine. And also, kidney also involved in regulation of blood pressure through RAS, renin-angiotensin system. When blood pressure is reduced, okay, what will happen? When renal blood flow is reduced, juxtaglomerular cell in kidney will convert the precursor prorenin into renin. So plasma renin then carries out the conversion of angiotensinogen released by the liver to angiotensin 1. Angiotensin 1 is subsequently converted to angiotensin 2 by the angiotensin converting enzyme, also called as ACE. Angiotensin 2 is a potent vasoconstrictive peptide that causes blood vessels to narrow, resulting in increased blood pressure. Angiotensin 2 also stimulates the secretion of the hormone aldosterone from the adrenal cortex. So aldosterone causes the renal tubules to increase the reabsorption of sodium, which is consequence causes the reabsorption of water into the blood, while at the same time causing the excretion of potassium to maintain electrolyte balance. So this increases the volume of extracellular fluid in the body, which also increases blood pressure. Okay, second, types and structure of nephrons. There are two types of nephrons, cortical nephrons and juxtaglomerular nephron. Cortical nephron, which are located in renal cortex, and juxtamedullary nephron lie in renal cortex close to renal medulla. Cortical nephron. So the renal corpuscle of the cortical nephrons located in superficial region of the cortex. The glomerulus is usually smaller. Loop of Henle is short and does not have a thin ascending limb. And the juxtamedullary nephrons, the renal corpuscle of juxtamedullary nephrons located in the juxtamedullary region, which is in the inner region of the cortex, which close to the medulla. The glomerulus is usually larger than the cortical nephron, and they are having long loop of Henle. Also have thin ascending part of loop of Henle. So here I made up in this table for the differentiations of juxtamedullary nephron and cortical nephron. in terms of its renal corpuscle, glomerular size, loop of Henle, main function, and efficiency in concentrating urine. So for the juxtamedullary nephron, its renal corpuscle located in the juxtamedullary region, while the cortical nephron located in the superficial layer of cortex. For the glomerular size, for juxta, it's large, while for cortical, it's small. Loop of Henle for juxta is long and has thin ascending limb and extend deep into the inner medulla. Dia ada ascending limb daripada thick kemudian kepada thin, dan dia extend deep into the inner medulla. While for cortical nephron, sebab dia duduk di cortical, so it's small, does not have a thin ascending limb and does not extend into the inner medulla. So the main function juxtamedulla is for urine concentration, while cortical nephron, urine formation and excretion of waste product. Efficiency in concentrating urine, juxta is more, while the cortical nephron is less. So our next microstructure of filtration barrier. So the formation of urine in the kidney involves three major steps: ultrafiltration, tubular reabsorption, and tubular circulation. You will learn later from other lecturers and other lecture. So, ultrafiltration is the first step in urine formation, occurs in the renal corpuscle, in which plasma is differentially filtered to form the tubular fluid. This is the initial and important step of urine formation, as it considerably contributes to the volumes and composition of the urine. So there are filtration barrier. So we will have into its structure of the filtration barrier. The filtration barrier consists of three components: endothelial cells from glomerular capillaries, glomerular basement, and epithelial cells of Bowman's capsule, also called as podocytes. So we can see here the glomerular capillary endothelial cell, the red color, and then neighbor with the basement membrane, and next to it is podocytes. Podocytes which has the food process. These three form the filtration barrier. So we look into more detail into its structure. So microstructure of the filtration barrier. For the capillary endothelium, capillary endothelium is fenestrated, surrounded by the glomerular basement, along with specialized cells called podocyte. Okay, capillary endothelium. Here the red one, endothelial cell, and then there is basal lamina and the podocytes. So the basement membrane, we go more into the capillary endothelium. The capillary endothelium ni fenestrated, as it contains fenestra. Fenestra means pore or windows. So they put in line and has some windows lah in between, which make up the filter. So the pore diameter is about 50 to 100 nanometer, and therefore they freely allow passage of large molecules through them. This allow water and small solutes like sodium, urea, and glucose to pass through the capillary endothelium. The endothelium cells are negatively charged due to presence of anionic glycoproteins on their surface. Therefore, protein molecules are not easily filtered through the filtration barrier. Hence, there is no protein in our urine. And we see here the basement membrane, also called as basal lamina. The basement membrane is made up of a meshwork or network of fine fibrils which entrench in the gel-like matrix. It is an important barrier for the proteins because the matrix contains collagen, laminin, fibronectin, and other negatively charged protein. So they not allow proteins to come in. So here is the histological slide for filtration barrier. There are fenestrae, endothelial cell, basement membrane, and there is a slit diaphragm and food process. Okay. Okay, we go deeper into the food processes of podocytes. The podocytes or food cells form the third or the inner layer of the filtration barrier, because we have three, like, so the food process is the third one or the inner. So the podocytes constitute the visceral layer of Bowman's capsule. There are also epithelial cells, and the extensions terminate as food processes that lie on the outer layer of the basement membrane. So dia mengunjur, memanjang sampai ke basement membrane. Dia macam kaki lah. Kaki dia memanjang. Dia pun pijak atas ni. Tapi dia ada some space which will call as slit pore or filtration slit, about 20 nanometer. The filtration slits are bridged by a thin diaphragm called a filtration slit diaphragm that contains small rectangular pores which do not allow free passage of molecules. So, dia ada 3 level of filtration. So, for conclusion, our filtration through glomerular capsular filtration barrier occurs mostly through extracellular route that consists of pores in the capillary endothelial cell layer, basement membrane, and slit pores in the food processes of podocytes of Bowman's capsule epithelium. In diseases like nephrotic syndrome, the permeability of the glomerular capillaries to proteins increases that leads to proteinuria. Okay, one more thing is the food ni dia interdigitate. We call it as interdigitate. Dia saling berganding. Okay. Messenger cell. Okay, there are also messenger cell. Kalau kita perasan kan, messenger cell ni provide messenger cell ni from blood monocytes and from the component of mononuclear phagocyte system. So its function is to provide structural support for the glomerular capillary and secrete prostaglandin and cytokines, and also phagocytic. Messenger cells that are present outside of the glomerulus, called as extraglomerular messenger cells or Lucky cell or Gourmati cell. They secrete renin and erythropoietin and also possess phagocytic activity. And besides, there is also apparatus called juxtaglomerular apparatus, which form from macula densa, juxtaglomerular cell, and Lackey cell. The juxtaglomerular apparatus ni controls the rate of glomerular blood flow depending on various factors such as hydration, blood flow, blood volume, and iron concentration. So the juxtaglomerular dia ada macula densa and juxtaglomerular cells ni. Juxtaglomerular apparatus ni quite important lah, sebab dia ada function of sensor. Okay, So the most important part in the juxtaglomerular apparatus ni, macula densa. Macula densa is modified epithelial cells of the thick ascending limb of the lobe of Henle when it comes in contact with the afferent and efferent arterioles. Kan dia bertemu, so it appears as a dense area. So that's why it gets the name as macula densa. So it acts as the sensor that monitors the change in ionic composition and rate of flow of the tubular fluid in the lumen of the tubule. Dia adalah sensor. So the macula densa cell sense the sodium chloride concentration in the lumen and stimulate the juxtaglomerular cell to synthesize and release renin into the bloodstream. This happens in states of hypovolemia or low sodium chloride concentration in the blood and in the urinary filtrate. This will activate the renin-angiotensin-aldosterone system and trigger several key important steps to improve blood pressure. The release of renin in the bloodstream causes hydrolysis of angiotensinogen to form angiotensin 1. Angiotensin 1 is converted by angiotensin converting enzyme to angiotensin 2. Angiotensin 2 will trigger the adrenal glands to produce and release aldosterone, which acts on tubule to upregulate sodium and water reabsorption back into the blood. So we also have Lacus cell. Lacus or Gourmati cell. Gourmati cell present in the triangular space formed by efferent and afferent arterioles and the macula densa. So, Lacus cell ni secretes some quantity of renin and erythropoietin. Another function of juxtaglomerular apparatus ni, juxtaglomerular apparatus secretes renin, which activate renin-angiotensin system, which is involved primarily in the regulation of blood volume and pressure. Second, macula densa cells act as sensor that detects the change in the rate of flow and volume of flow in the tubule and composition of tubular fluid, which provide feedback signal to the glomerulus to change the rate of filtration, which forms the physiological basis of tubuloglomerular feedback. Thus, they control glomerular filtration. And Lacus cells secrete renin and erythropoietin. So the last is renal circulation. Okay, special thing about kidney receives about 23.5% of the cardiac output, though they constitute less than 0.5% of total body weight. So kidney receives quite a big amount of of the blood flow to kidney. Okay, the blood flow to kidney is about 1,260 ml per minute or 400 to 20 ml per 100 gram of tissue per minute. Thus, blood flow per unit weight of the kidney tissue is much more in comparison to other organs. Nak cakapnya, kidney ni terima innovation yang banyak dan juga blood flow yang agak besar compared to other organs. So, however, there is differences lah in the regional distribution of renal blood flow, with flow being maximum in the cortex and minimum in the medulla. Medulla about 0.5, cortex about 5. Okay, so here we see the chart of diagram of renal blood flow. Start from the aorta. Okay, for this renal circulation ni, Each kidney is supplied by a single renal artery that originates from aorta. Okay, first, renal artery branches to form anterior and posterior divisions that form total 5 segmental arteries. Each segmental artery branches into interlobar artery, which is successive divisions form arcuate artery, interlobular artery, and afferent arteriole. So afferent arteriole give rise to glomerular capillary network that finally drains to efferent arteriole. Efferent arterioles then lead to a second capillary network, which is formed around the renal tubule, which we call it as peritubular capillaries. Okay, the venous system also parallel to the arterial system. So the efferent arteriole form peritubular capillaries or vasa recta, which drain into interlobular veins. The subsequent sequence of drainage is arcuate vein, interlobar vein, segmental veins, and finally the renal vein. And in the medulla of kidney, blood supply is derived from afferent arteriole of juxtamedullary glomeruli. This efferent arteriole in juxtaglomerular nephron, in addition to formation of peritubular capillaries, form an extra set of capillaries called vasa recta. We'll go to vasa recta later. Vasa recta are straight and long capillaries that run deep into the medulla. And this arrangement of vasa recta helps it to function as the counter exchanger in urine concentrating mechanism by exchanging substances between blood flowing into and out of medulla. So vasa recta is important, provides oxygen and nutrients to the nephron segments, deliver substances to nephrons for secretion into tubular lumen, serves as a pathway for the return or reabsorbed water and solutes to the circulatory system, and participate in concentration and dilution of urine. Importance of renal blood flow supplies oxygen, nutrient, and hormones that control kidney function. And blood flow also deliver metabolites and waste products to kidney for their excretion in the urine, and control concentration and dilution of urine, influence solute and water reabsorption, determine glomerular filter rate. Okay, now for the renal circulatory, I bring you someone to explain here.
The nephrons of the kidney have a unique blood flow. So if one were to trace the flow of blood to a nephron and back, say beginning with the abdominal aorta, one would follow the renal artery as it enters a kidney, and the renal artery then branches into 5 segmental arteries. And in the renal pelvis, these segmental arteries will divide into several interlobar arteries. These interlobar arteries form arcuate arteries as they arch over the pyramids in the renal medulla. And from these arcuate arteries, cortical radiate arteries then approach the cortical tissue. Afferent arterioles depart from these and enter the renal corpuscle, forming the glomerular capillaries. Now, the glomerular capillaries are drained by efferent arterioles. Nowhere else in the body are capillaries drained by an arteriole. These efferent arterioles then form a second capillary network, either the peritubular capillaries around the PCT and DCT, or the vasa recta, which surrounds the loop of Henle. These capillaries then drain into cortical radiate veins, which drain into arcuate veins, which drain into interlobar veins, which drain into segmental veins, which drains into the renal vein, which departs the kidney and brings the deoxygenated blood to the inferior vena cava. The
Okay, so we reach to the last subchapter of regulation of renal blood flow. So there are a few ways to regulate renal blood flow, which are neurofactors, hormonal factors, local factors, and autoregulation. Okay, for neurofactors, kidneys receive a rich supply of sympathetic nervous system that enter the organ alongside the blood vessel. So stimulation of renal sympathetic fibers causes vasoconstriction that decreases renal blood flow, and conditions to activate renal sympathetic fibers such as hemorrhage, cold, pain, exercise, and anesthesia. And stimulation for a longer duration as occurs in hypovolemic shock, iaitu when people lose about 20% of their body's fluid or blood supply, will cause renal shutdown and exothermia. Exothermia means elevation of blood urea nitrogen and serum creatinine level. And hormonal factors, catecholamines and other vasoactive. Okay, hormonal factors, there are two main hormone which are catecholamines and other vasoactive peptides like angiotensin 2, which cause renal vasoconstriction, and dopamine in reverse in high dose causes vasodilation. Therefore, dopamine is preferred for the treatment of cardiogenic shock, as its systemic effect increases blood pressure and renal vasodilation effect maintains renal perfusion. Ni for example lah. So hormonal factors also contribute to the renal blood flow regulation. Besides a part of outer factors, renal also have their own intrinsic system, which we call as autoregulation. So renal blood flow autoregulation is a vital homeostatic mechanism that protects the kidney from elevation in arterial pressure. So dia ada their own sistem sendiri yang boleh mengawal blood pressure, arterial pressure. So it involves two mechanisms, which are myogenic mechanism and also tubuloglomerular feedback. So from the high blood flow to kidney, which increase pressure in blood vessels, and this will stretch the vessel wall of afferent arterioles and cause opening the cation channel, which results in depolarization. So it will lead to voltage dependent opening of calcium channel, causing the calcium influx. So calcium masuk. So increase intracellular calcium will cause vasoconstriction, which results in low blood flow to kidney, back to normal. And the tubuloglomerular feedback involves the concept of purinergic signaling, in which an increased distal sodium chloride concentration cause a release of adenosine from the macula densa. This initiates cascades of events that ultimately brings GFR to an appropriate level. Okay, we end here. Okay, I think that's all for today. Thank you very much to all of you. Inshallah, see you later. Assalamualaikum and thank you very much.